We're trying to get started. So, hello everybody. Welcome to BrainMap. Before we start, just a reminder that the seminar series is co-sponsored by P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. With that out of the way, let me introduce our speaker for today, Dr. Pedro Brugarolas. Dr. Brugarolas is an assistant professor in the Department of Radiology at MGH, HMS, and is a member of the Center of Nuclear Medicine and Theranostic Sciences and the Center for Interdisciplinary Innovation in Imaging. He's a radiochemist and neuroimaging scientist whose research focuses on developing novel PET tracers to probe molecular mechanisms of neurological diseases. Dr. Brugarolas is a developer of 18F3FAP and leads a research program aimed at creating imaging tools to assess demyelination, is that correct? Demyelination and neuronal dysfunction and therapeutic response across a range of disorders. So with that, let's welcome our speaker.
Thank you, Dinan, for the kind introduction, and thank you very much for the invitation. It's really an honor to speak to this crowd, and I want to tell you about our work with 3F4AP, and I hope that by the end of the talk, I convince you that mapping the brain with 3F4AP PET can play a big role in mesoscale imaging of the brain. So let me start by introducing my favorite protein. Here is the crystal structure of Kv1.2, also known as KCNA. This is a voltage-gated potassium ions, and it's a remarkable example of molecular machinery. These allowed the selective passage of potassium ions from inside of the cell to outside of the cell, and it's selected for potassium even though you have other ions like sodium but have the same charge and are smaller, but this can discriminate between the two. And this is going to be the target of the tracer that we develop. They are present on every excitable cell in your body, and then in neurons, they play an important role in propagation of action potentials. So, as you know, a neuron gets an input, and then if it decides to fire an action potential, you have sodium channels open first, and then sodium rushes out from outside of the cell to inside of the cell, and that depolarizes the membrane. It goes from a resting potential of minus 70 millivolts to plus 40 or so millivolts. And then when the voltage reaches that level, then potassium channels open. There's a voltage sensing domain, which is not shown on this structure, but that opens for potassium channels, and then you have repolarization. So potassium channels rush from inside of the cell to outside of the cell. And so on normal unmyelinated axons, you have potassium and sodium channels throughout the whole axon, and they're working in tandem. Sodium channels open, potassium channels open, and that propagates the action potential. On myelinated axons, you have the sodium channels are concentrated at the node of Ranvier, and the potassium channels are flanking the node immediately underneath the myelin. So here on this scheme, you can see in the node of Ranvier, which is the gaps between the myelinated segment, you have high concentration of sodium channels, and then at the juxtapar node, you have high concentration of potassium channels. When there is demyelination, now your potassium channels, they sort of spread out through the demyelinated segment. And that's what causes demyelinated axons to not be able to propagate action potentials. So here's a confocal image of neurons in an optic nerve of cuprizone-fed mice. So these mice develop severe demyelination. And you can see on a control mouse, the sodium channels are clustered at the node, and the potassium channels are on the side, shown in green. When you have demyelination, both the sodium channels and the potassium channels, they spread out. And there's a pretty substantial increase in expression, somewhere between 2 to 10-fold in the demyelinated segment. And this has been shown almost in every model of demyelination and also in humans. So going back to the structure of the potassium channels, there is here in the intracellular side of the pore, there's a cavity, and that is where 4-aminopyridine, which is this very simple small molecule, binds to. So 4-aminopyridine is also known as dalfampridine, is an FDA-approved drug for multiple sclerosis. So it doesn't look like a drug. It is very small. It doesn't have enough hydrogen bond donors and acceptors. But the way it works so well is under physiological pH, this molecule is mostly neutral, and then it can diffuse through biological membranes, including the blood-brain barrier. So it gets into the brain, it gets into every cell, and then, but then there's a small fraction that is in the protonated form, which then gets into the cavity of the channel. It gets stuck. It's too big, it cannot go through. So the idea that we had was, can we use this molecule to detect potassium channels and changes in potassium channels due to demyelination? So we proposed that perhaps putting a fluorine 18 isotope on 4AP would work to image demyelination. So before I go into the development of the 4AP, let me tell you why imaging demyelination by PET. So demyelination, as you know, is common in many both CNS and PNS diseases. So of course, we'll know about multiple sclerosis. It's an immune-mediated demyelinating disease, but demyelination also happens in multiple genetic diseases. It happens after traumatic conditions like TBI, traumatic brain injury, or traumatic spinal cord injury. It happens in neurodegenerative diseases like Alzheimer's disease and stroke. There's some reports that demyelination may be one of the early events in Alzheimer's disease. And in the peripheral nervous system, demyelination can happen from infections. After an infection like Guillain-Barré, it can happen from hereditary neuropathy. It can happen with diabetic neuropathy. There are many, and MRI does a pretty good job at detecting demyelination. So obviously myelin is very lipid rich. So when myelin is, and that gives a lot of MR contrast, but it's not very quantitative or specific. So here is where a marker, a PET marker for demyelination can help. We're not the only ones thinking about imaging myelin or demyelination by PET. Some of the first attempts to image demyelination are from before I was born, with these very lipophilic radiolabeled molecules like iodobensin. These are, yes, compounds that get into the brain and they stick to lipids. And when MRI came along, I think the use for these molecules, it sort of went away. But there was a resurgence with the development of the tracers for amyloid, for the tracers for AD, because if you have looked at amyloid PET images, there's some binding to the white matter. There's some non-specific, potentially specific binding to myelin as well. So we some of these amyloid agents, people have explored their use for imaging demyelination. And then, so here is a preclinical study using 3 different tracers. PIB is a well-known amyloid tracer. CIC and MEDUS are derivatives with a steel beam core. And this is a model of chemically induced demyelination. So they inject a surfactant into the brain that causes focal demyelination, and you can see a decrease in signal. I would argue that it's not ideal to image a decrease in signal by PET. Ideally, you want to increase in signal. So rather than targeting myelin itself, you want to target a protein that is present in demyelinated axons. Nevertheless, this has been evaluated in people with multiple sclerosis and is able to quantify changes in myelin pretty accurately. So now going back to our approach. So we proposed that this fluorinated 4-aminopyridine derivative could potentially be a good PET tracer for imaging potassium channels. So the first thing that we had to do is to make sure that when you put a fluorine in this molecule, it still binds to the channel. And we did that using a functional electrophysiology assay So here you have potassium Kv 1.2 expressed in frog oocytes, and you're applying a voltage, and then when you apply a voltage, the channels open and potassium goes through. You have a medium that only contains potassium. When you give 4-AP, it blocks the channel and you don't see the current going through. 3-4-AP, that's the same thing. So this confirmed that it's binding to the channel with about the same efficacy. Then we purchased the carbon-14 labeled form of this molecule to do some autoradiography. And the way this autoradiography was done is we injected the carbon-14 labeled compound to live animals that had a myelin deficit, like the shiver mouse. The shiver mouse has a mutation in myelin basic protein and does not produce myelin. And what you can see on the histology staining, on the LFB staining, is that the shiver mouse has the corpus callosum looks sort of much more faint than on the control mouse, where you have a lot of myelinated axons. And then 3-4-AP showed low signal in the control mouse and high signal in the demyelinated mouse. We confirmed this in 2 other models. This is a model of demyelination followed by remyelination, and you can see that the signal goes up and then goes down upon remyelination. And this is a model similar to the one I showed before of chemically induced demyelination, and we can see a hot spot in the area of demyelination. So once we had that, we set out to radiolabel this with F18. The standard methods for F18 labeling, like copper-mediated fluorination or boronic esters or iodonium ylides, did not work for this compound. So we had to come up with another way to do it. The first method that we developed was using a pyridine oxide. By putting the oxide, that makes this molecule more prone to aromatic nucleophilic substitution. So we were able to put the F18 in there, and then you have to remove the N-oxide, which is done using hydrogen gas over palladium carbon, which is not very automation friendly. So this was good to do some pilot studies, but it's not ideal if you want to do studies in animals and humans. So we then, in collaboration with folks at the NIH, we developed this other method that uses a nicotinic acid derivative. And this proved to be pretty robust and reproducible. And it takes about 90 minutes and produces around 15% radiochemical, non-decay corrected radiochemical yield. So this is one of the first experiments that we did. Again, going back to the model of chemical induced demyelination, control animals receive an intracranial injection of saline, and we don't see much on the PET. Demyelinated animals, we see an increase on the PET. This was confirmed by post-mortem autoradiography, which matches very well with the histological staining for myelin. So where you see the demyelinated area, you also see the high signal on autoradiography. So at that point, we wanted to bring this tracer to human study. So the first step was to validate it in non-human primates to make sure that it had adequate kinetics for human brain imaging. So with my colleague Mark Normandin, we did long dynamic scans in multiple monkeys. And then during the scanning, we sampled arterial blood, we measured metabolites and, for kinetic modeling. For those of you who are not very familiar with kinetic modeling, essentially the scanner gives you an image that is the sum of the tracer that is bound to the target, plus the tracer that is floating around that is not bound to anything, plus the tracer that is not specifically bound to lipids and other structures, plus whatever is in the blood pool in the brain. But if you measure the concentration in the whole brain, and you also measure, or in any brain pixel, and then you also measure the concentration in blood, you can write differential equations and solve for these different compartments. And then, because at the end, what you want is to know your volume of distribution, which is proportional to the concentration of the receptor. So we measure the concentration in blood. It looked pretty consistent across animal. We measure metabolites, and it looked remarkably stable. After 90 minutes post-injection, there was less than 10% metabolism. Some time activity curves in the brain of 2 monkeys. They look pretty consistent with each other, and then during repeated scans, the standard deviation was pretty narrow, and so we felt pretty good that this had good kinetics to continue to move it forward. But we found one surprise, and it's one of the monkeys showed this very bright spot in the frontal cortex on the left side, and we had never seen this before. This monkey had been scanned with many other tracers, and they had never noticed anything unusual there. And you can see that the kinetics of what we call the lesion were very different than the kinetics in the rest of the brain. The initial peak is lower, meaning it's hypoperfused, but then the washout is a lot slower, meaning there's more binding. So looking a little bit closely at the images, we noticed that on the CT, there was actually a hole in the CT. So this monkey had belonged to a different investigator who wanted to put an electrode in the brain and use it for electrical stimulation studies. During the procedure, they damaged the brain. There was some bleeding. They removed the animal from the study. And then after it had healed, my colleague adopted the animal for pet studies, thinking that it was a healthy animal. And this was 3 years after the procedure happened. We looked at other tracers that we were working with at the time. PBR20, so 3F4AP showed very bright signal in the lesion. PBR28 showed a small decrease in the lesion. You wouldn't necessarily expect to see a lot of inflammation 3 years after the injury. The PIP didn't show any major changes, and FDG showed some signal outside of the brain, but not in the brain. And here, when we look at the quantification of the different brain regions, what is interesting with 3F4AP is all the brain regions are within 20% of the mean of the brain, but the lesion is 6 or 7 standard deviations away. There's a big increase. There's a 40% increase. So the healthy brain is pretty narrow dynamic range. And then 3F4AP showed these very high increases in signal. All the others show a modest decrease in signal. This animal expired a few months ago, and now we have the brain in a jar. So I'm looking forward to doing additional studies in the postmortem brain histology, postmortem MRI, to see what is going on there, whether this is due to demyelination or due to other possible reasons. Unfortunately, this was a one-off monkey. We could not easily replicate this study for humane reasons, but also timing and cost consideration. This was 3 years after the procedure. So we decided to look into a model of penetrating TBI in mice. So we teamed up with Eng Law, whose lab had a model of controlled cortical impact. Here is a similar model in that you cut a piece of the skull and then you do a control impact in the cortex. And when we image these animals, they showed a very bright hot spot on the head, which was not present on the sham animals. And then we did kinetic modeling on these animals. So we obtained an image-derived input function from the heart. So here is the blood curve. Here is the contralateral side, and here is the injury side. Again, you can see a lot of lower washout, much greater binding in the TBI than in the sham. And one thing that was good from this study is we could do the quantification using SUVR, and that we showed that that was very proportional to DVR, which is more absolute quantification. So a post-mortem histology on these animals confirmed that there was demyelination. Here you can see the cavity from the impact, and there's some demyelination in the corpus callosum. Whether there's other contributions to other potassium channels that are contributing to this signal is something that we're still want to investigate. We followed these animals for over a month, and we can see that the signal appears as early as 3 days, it peaks at 7 days, and then it slowly goes away. And of course, the sham, we couldn't see anything on the sham animals. So next, we wanted to see whether this would apply to closed head injuries, which is much more relevant to human TBI. Most TBIs are not penetrating TBI. So we looked for someone who had characterized a method of closed head injury, and Regina Armstrong had published a method showing that when you do an impact on the closest skull, that causes demyelination in the white matter. So she sent us some animals, and we did PET on these animals. And in the closed head injury, we could also see it was not as large the effect as we saw in the open head injury, but there was about a 10 or 15% increase in signal. And this is even in the absence of overt changes in the histology. So moving down from the brain to the spinal cord, we decided to look at whether spinal cord injuries would also show up with this tracer. And in the spinal cord, it had been well documented that potassium channels, they increase in expression several fold, and they disperse through the injury site. So we team up with Stigand He at Boston Children, whose lab had this model in-house. They created a control impact into the spinal cord. So essentially, they do a surgery to remove one of the vertebral bones, and they do an impact on the spine. And there was very high signal there in the area of the injury. After the PET, we dissected the spinal cord and did also radiography on the whole cord, and you can see very high signal and very focal. So the signal in the injury is more than twice the signal in the adjacent segments. And you can see that the kinetics, they are consistent with what we had seen on the TBI mice. We were able to follow a cohort of animals for out to 1 month, and what we saw is that the signal peaks around 7 days and then stays high, and it actually sort of disperses rostrally and caudally on the spinal cord, which is consistent with demyelination. This model also provided the opportunity to look a little bit closer into where it was binding. So here, if we look at panel B first, you can see that the white matter tract shown in blue, there is demyelination, and those are the areas that show high signal on its radiography. So the signal is not coming from the cavity, it's actually coming from the area, from the demyelinated white matter. And the experiment here on A, it shows that the binding is pretty specific, because if you do ex vivo radiography, meaning you inject the tracer when the animal is alive, and then dissect the tissue, it lyses it very quickly and do a radiography, you see this very focal signal in the injury. But if you take a tissue slice from the same animal the next day, and then you apply the tracer to the tissue, you don't see any binding at all. So if the binding was non-specific, you would expect that it would remain on the tissue, that you would still see binding to the tissue. But here there was basically no binding if you compare the signal to the background. So if we continue to go down from the brain to the spinal cord to the sciatic nerve, we decided to look at whether we could pick up peripheral nerve demyelination. So peripheral nerve demyelination happens after nerve injuries, but also after in multiple neuropathies and after infections. So we purchase animals with a spinal nerve ligation. This is a commercially available model from Charles River. They do a tight ligature on the L5 and L6 branches of the sciatic nerve. And then, so the first thing that we did was confirm that there was demyelination by Western blot. And you can see that on the non-ligated side, on the left side, MVP shows high signal on Western blot, myelin basic protein, and low signal on the injured side. We did multimodal MRI and PET-CT imaging. And so the MRI showed that the nerve was swollen, and there was also swelling in the muscle around the surgical site. And then PET showed very bright along the ligated nerve, but not on the contralateral nerve. And we were able to quantify about a 3.6-fold increase based on the PET. After the PET and the MRI, the animals were euthanized. We dissected the nerves. You can see the the ligatures in some of the branches. And then we did autoradiography, gamma counting, and histology. Autoradiography showed much higher signal in the injured side compared to the contralateral side, which was confirmed by gamma counting. And then the histology, these are toluidine blue fiber staining. The control nerve, the control side, showed that 97% or 99% of the large caliber fibers were myelinated, whereas in the ligated side, only around 37% were myelinated. There was also some axonal loss by counting all the axons in each nerve. There was about a 30% decrease in axon density, and which is interesting because you have demyelination, which is increasing your PET signal, loss of axons that is decreasing your PET signal, but overall the net signal is still highly increased. All right, so the preclinical animal studies were pretty exciting, but we wanted to move these to humans. So we set up a fully automated method. So the chemistry is the same as what I showed before, but the initial automation was done on an NAPTIS module. It turned not to be very reliable. So most recently, we have switched to an FX2N module, and we got an IND a few years ago, and we started doing human studies. Here is an image from the first volunteer. And this is one of the early images. I think this is from the first 30 minutes. You can see pretty high signal in the brain, and then high signal in some of the internal organs like the kidneys and the urinary bladder. You can see the vein in which it was injected. And based on this data, we were able to calculate dosimetry. The effective dose was 12 microsieverts per megabecquerel, which is comparable to other F18 tracers, maybe a little bit lower than FDG. We looked at the kinetics of the tracer in the brain, and this was interesting because so in the monkeys we had seen that it peaked higher, but then it plateaued at a higher level around 1.5 or 2. In humans, it was washing out faster, and this is not typically what happens. Typically, when you go from monkeys to humans, things slow down. They don't speed up. When we look at the blood metabolite, what we saw is that in monkeys, after 90 minutes, you still had 90% parent. So these are stack HPLC chromatograms. The parent is showing here around 10 minutes on the chromatogram. But in humans, we were seeing only around 20% remaining 90 minutes post on the 90-minute blood sample. So this didn't make much sense to us. We had also looked at metabolism in mice, and mice showed the same behavior as humans, where the tracer was not getting metabolized. So we thought, well, could this be due to the anesthesia? So monkeys are imaged under anesthesia, humans are imaged awake. Could anesthesia be causing these drastic differences in metabolism? So we thought of scanning awake monkeys, but that didn't sound very safe. I've seen a rhesus macaque wake up during a scan, and it's not pretty. We thought of scanning humans anesthetized, but that would have been hard to get it through the IRB. So we settled on a third option, which was doing a comparison between anesthetized mice and awake mice. So we took two groups of mice. One group received the tracer and were kept under anesthesia. The other group received the tracer while being physically restrained, and they were free to move around. And then what we saw was, again, the signal in the brain in the awake mice was a lot lower than the signal in the anesthetized mice. And this was consistent with what we had seen on the awake human primate compared to the anesthetized non-human primate. When we look at the metabolism, both in the plasma and in the brain, we saw that anesthetized animals hardly metabolize the tracer, whereas awake animals, they extensively metabolize the tracer. Luckily, it looks like there aren't many metabolites that are getting into the brain, so this does not pose a quantification challenge. The peak that we see here, we think that's just the blood pool from the brain. We dove deeper, try to understand what the metabolites produced were. So we generated multiple deuterated derivatives of 3F4AP with deuteration in different positions and different percentages. And then using a combination of an enzymatic assay and mass spec, we were able to determine that the metabolic products are either the formation of the N oxide or the formation of the 5-hydroxypyridine, but not the 2 or 6-hydroxypyridine. Then going back to the human studies, we have done a couple of small studies, one in people with multiple sclerosis and one in people with a spinal cord injury. I'll show the data from MS first. So here's a healthy control. The CT, the MRI, and the PET, the early PET images, they yet look like blood flow images. The late PET images on a healthy subject, they look pretty homogeneous. Again, the time activity curve for the brain. And then, so here is some data in MS. And this was a small study with 3 healthy controls and 3 MS. MRI myelin water fraction shows high myelin density on the control subjects and lower myelin density on the MS subjects, as you would expect. The PET shows also very remarkable differences. The PET images from MS are brighter than the ones from the healthy controls. You can also see some focal spots. Those are MS lesions. And this is visible both on late SUV images, but also on parametric images using Logan VT. The quantification showed that even with a small sample size, we could detect differences in VT between the MS and healthy controls in different brain regions. The Logan VT and 2-tissue compartment modeling VT, they correlated very well. And then 2-tissue compartment VT and late SUV, they correlated well at the individual level and at the subgroup level. At the whole level, there wasn't that good of a correlation. We need to look more into it. But I want to show the images from one of the participants. And this is a T1 MPRH, and this is a T2 flare from a person with MS. On the MPRH, you see a number of periventricular lesions. They look dark on T1. None of them are gadolinium enhancing. And then on T2, they are all bright. If you look at the 3 lesions that are marked by the arrows, they look pretty similar to each other. But then when you look at the PET, one lesion is clearly bright, one is dark, and one is in between. And this is sort of what we had hypothesized, but we still couldn't believe it when we saw it. So what we think is happening is when you have normally myelinated axon, in this scenario, you have low levels of PET tracer binding because your potassium channels are masked by the myelin. When you have demyelination, you have high levels of PET tracer binding. But after a while, neurons that are demyelinated, they will either remyelinate, and then the PET signal will go back to baseline or near baseline. So that's what we think is happening here. This lesion is remyelinated. If they remain demyelinated for a long time, they may degenerate. You have axonal loss. In that case, your PET signal will drop. So what we think is this is telling us differences between these lesions where one has a lot of demyelination, the other, the axons are lost, and the other, it has been remyelinated. And this is different from what you see with myelin water fraction on MRI. Myelin water fraction in healthy controls show a moderate correlation with the PET signal. but in MS, it did not show a good correlation. So again, myelin water fraction and 3-4-AP PET are giving you different information. More recently, we scanned a person with MS that was newly diagnosed and treatment naive. And this person had several gadolinium-enhancing lesions. So people under treatment usually don't have many gadolinium-enhancing lesions. These lesions showed us incredibly bright on the PET. You could see it a mile away. Here are the time activity curves for the white matter in red and for the lesion. And the increase was several-fold increase. So again, you can see that the healthy brain regions are down here, and then the injured or the lesion, the gut-enhancing lesions are up here. This person also had some gray matter lesions, which are hard to see on MRI, but they could also be picked up on the PET. And we could tell that at least for the gut-enhancing lesion, it was not due to a change in perfusion, but due to a change in binding based on the kinetic modeling results. So now looking at individual participants, so these are the first 3 people with MS, and here's the person with the newly diagnosed person. What we see is that there are 2 or maybe 3 types of lesions. You have lesions that are bright, so the lesion is significantly higher than the paired contralateral mirror ROI. And then there are lesions that are essentially the same level. Those we think are remyelinated. And then MS3 was interesting because this was a person with progressive disease. And here you see a lot of lesions that have this downward trend, meaning there are dark lesions, which we think there's a lot of degeneration. On the person with the gut-enhancing lesions, we saw a lot of bright lesions. On the ones that were non-gut enhancing, the ones that were gut enhancing were very, very bright. And then the gray matter lesions were somewhat brighter. And I think I'm coming to the end now. Here is the data in a person with a severe spinal cord injury. This person, you can see here, they have a metal plate supporting the spine, and there was a crushed vertebra here. This person initially was diagnosed as a complete injury and later recovered some function, but he was still wheelchair bound. And what you can see is that below the injury, the signal in the cord is very dark, it's very low. In the initial PET frames, this suggests low perfusion to that area. And then in the late PET frames, it didn't seem elevated or reduced. This was very different from what we saw on another person that had a milder spinal cord injury. This was a person that had a spinal tumor that was removed. When the tumor was there, it was pressing against the spine and the person was paralyzed. When it was removed, it recovered almost completely. And what we see here is that on the early PET images, we see some bright spots, potentially indicating hyperperfusion and maybe inflammation. And then on the late PET, we saw a slightly elevated signal, possibly indicative of demyelination. So I want to end with a sort of a what comes next slide. These are, yes, a quick search of potassium channels and diseases. And we've been looking at changes in KB channels due to demyelination, but KB channels are involved in many other diseases. Mutations in KB channels cause epilepsy and cerebellar ataxia. They're also involved in Alzheimer's disease and other degenerative conditions. They're also in the cardiovascular system and in immune cells. And these are some of the current uses of 4-aminopyridine. So it's used for MS, but it's also used off-label for spinal cord injury. There's people that want to use 4-AP for traumatic brain injury. It is used off-label for neuromuscular diseases. So we think there's plenty more to investigate with this tracer. And now this tracer is available at Martinos. So I want to summarize by saying that 3-4-AP is based on an FDA-approved drug called dalfampridine, 4-aminopyridine. It binds to voltage-gated potassium channels, which are highly abundant in the brain. It's labeled with F18, so that has some advantages for doing PET, the longer half-life. We have method validated both on main campus and Martinos. It's brain permeable. It has a minimal plasma protein binding. It's pretty metabolically stable, and there are no brain penetrant metabolites. It has robust two-tissue compartment modeling kinetics with low variability. It has a narrow dynamic range in the healthy brain, but it showed large increases in pathology, and it has shown sensitive to changes in MS, TBI, spinal cord injury, and peripheral nerve injury. With that, I want to thank my current lab members and my former lab members who did a lot of this work, and then also a large network of collaborators. My lab works with many other labs. We don't prepare our animal models. We rely on other collaborators to prepare the models. I also have clinical collaborators that are finding the subject for human scans, and a number of extramural collaborators. I'm also grateful to the MGH PET core, which established the synthesis of this tracer, and more recently the Martinos PET core, which also established the synthesis here and will provide much needed redundancy. And I thank these agencies for their funding. At the moment, we're struggling for funding, but I'm hoping the situation will change, will turn around soon. With that, I thank you for your attention, and I'm happy to take any questions.
Okay, I think it's